You just have to start working on it. Thalamic problems, thalamic lesions are not rare, so you're going to be encountering them. The anterior nuclear group, in general, people don't divide up into many nuclei. It's just called the anterior nucleus by many people. All right, sits at the anterior portion. We have this lamina that divides the thalamus into components. And situated within it, we have the intralaminar nuclei. Here we have one, there we have one. When we go medially, we've got this medial group, the midline thalamic nucleus here, and the medial nuclear group, or the mediodorsal nucleus here. So you've got those there. Then we have, let me save this area for last. At the back end, so if this is anterior, this is the posterior group, and it consists of three structures: the pulvinar and the two geniculi, the medial geniculate and the lateral geniculate. Geniculate literally means knee, and those are the little knees at the back of the thalamus. The lateral group here is absolutely mind-bogglingly confusing. All right? The names are confusing until you sit down and think about it. So let's start out with the two superior ones. All right? We've got the lateral dorsal and we've got the lateral posterior in here. Okay? This one, that one. And then we have these three: the ventral anterior, the ventrolateral, and the ventral posterior lateral. Oh. And then we have this little one that's tucked internally, the ventral posterior medial. So these two are ventral and posterior. This and this are lateral. Actually, we got a whole bunch of lateral in here if we want to make that argument. So there's a lot going on. And finally, and it's really too bad that they made the cut on this one and they didn't show it over here. You see this clear one? This clear one is really kind of wild. That clear one is the reticular nucleus. It covers the whole lateral aspect. They show it clear on here so you can see that it's overlying everything else. That is a wildly important nucleus. So if I was to organize these for myself, to study them, I would have the reticular nucleus, which is along the lateral surface. I would have the anterior nucleus. Then I would have the the lateral nuclear group. Lateral dorsal, lateral posterior, ventral anterior, etc., etc. Then you have the mediodorsal, the intralaminar, and the posterior group. So you just have to figure out what works for you in organizing them. This is something that you really should practice drawing out. This is a good one. Okay? You could draw it out as, a lateral view. You could draw a cross section, or you could do it however you want. This is a good one to know really well. So, the complexities of the thalamus. First off, you have two, right? They are bilateral. So if we were to, once again, draw that little cross-section of the brain here. We have the capsule signals that pass through there. And then we have the left thalamus and the right thalamus. Okay? That's really important to remember. As I said, pretty much, the majority of cortical inputs pass through the thalamus, and many inputs to the cortex pass through the thalamus. But there's a lot of other structures involved here. Anterior group. Mammillothalamic tract. Thalamus. What's it tied to? The mammillary body. The mammillary has inputs to the anterior nucleus. The fornix has inputs to the anterior nucleus. Okay. Where's the mammillary body? What is it part of? What? The hypothalamus. So automatically, right now, you've got hypothalamic inputs to the thalamus, you've got fornix inputs. Fornix, behavior, memory. Hypothalamus, all of your homeostatic mechanisms. Oh, all of a sudden the thalamus is becoming more important. Ventral anterior, ventrolateral, okay? A lot of times ventrolateral is going to be divided up into two sections. We'll talk about those in just a minute. Input from substantia nigra, internal globus pallidus, all right? Substantia nigra, it's part of the midbrain nuclear group. What's that called? No, peduncles? Peduncles are always fiber tracts. Okay? What is the one group of nuclei in your brain that truly are misnamed? So we have these groups of cell bodies peripherally. What are they called? Ganglia. We have groups of cell bodies in the brain. What are they called? What's the one group of cell bodies in the brain that has a confusing name? What's the group in the brain called blah blah ganglia? The basal ganglia? Oh no, sorry, that was painful drawing that one out of you. Right? Yeah. Either the basal nuclear group, basal ganglia, whatever. Substantia nigra and internal and globus pallidus are part of that. Now, the important thing about that is that these are involved with movement. Well, all of a sudden, and we're just talking about a little bit of the thalamus here. We have inputs involved with behavior, memory, homeostasis, and movement. Whoa, that doesn't leave much. Yeah. The deep cerebellar nuclei coming into the ventrolateral nucleus. These are involved with movement as well. The deep lemniscus, the spinal thalamic tract. Oh, that's easy. What's it going from and to? Okay, go with it sometimes. Just run with it. Yeah. This is a clearinghouse. This ties together a lot of spinal. It's really redundant to say spinal tracts, but a lot of spinal tracts and the thalamus. This one is a little different into the ventral posterior medial layer. Trigeminal. Oh, trigeminal lemniscus. What's that involved with? Trigeminal nerve. Okay, trigeminal thalamic tract. Oh darn, this is tying together your trigeminal and the thalamus. And then we have the gustatory inputs. So now we're adding special sensations. We have taste coming through here. All right, let's go to the medial dorsal up here. Amygdala, olfaction, basal ganglia. Amygdala, memory, fear. Olfaction, olfactory cortex. Has to do a lot, by the way, with your limbic system. And basal ganglia movement, once again. Coming to the intralaminar area here, we have the internal globus pallidus, reticular formation, and a whole bunch of sensory pathways. Finally, sensory pathways. Reticular formation is outrageous. Your reticular formation is involved with your states of awareness and arousal. We're going to come to those later on in the semester. And then we come to the posterior group, the pulvinar and the two geniculae. The pulvinar gets inputs from the superior colliculus. We're going to look at the superior colliculus shortly and talk about that. But the two geniculae are really kind of neat because the lateral gets inputs from the optic tract and the medial geniculate gets inputs from the inferior colliculus, which is associated with auditory inputs. So all of a sudden the geniculae are all the special senses. So there's a lot going on. Table 7.3 on pages 284 and 285 is critical for you to work with. I just think I would make special effort to work with that. Maybe one thing for you to do is to print out, this and have it in front of you as you have that table open. It's a 2-page table. It takes up about half the page on the lower half. So the ventral posterolateral nucleus, okay? VPL. Spinal thalamic tract inputs. Somatosensory cortex. Spinal input to the cortex. So the spinal tracts are coming up to VPL. From here, they go to the somatosensory cortex. It's relaying information. Once again, this is critical for somatosensation. All I've done, we're going to look at a series of slides like this. Basically, what I've done is I've brought out one nucleus worth of information and isolated it on the slide. So, ventral posterior medial, VPM. Trigeminal thalamic tract, somatosensory cortex, and taste cortex. So, once again, we're getting information from that trigeminal nerve area. Relays cranial nerve input to the cortex, taste input to the cortex. Bang. We think, we've all learned the cranial nerves, and we think about them very discreetly. But the reality is, when they're bringing in information, that information has to be interpreted somewhere. It's so nice for this textbook and all other textbooks to isolate nerves, isolate nuclei. And that's not reality. The reality is, for learning them, isolating them is critical. However, the reality is, signals get into you, and it's not like the signal goes from, my eyeball to my optic cortex, whatever. No, it bounces all over the place it's interpreted, and